So we just finished talking about taxonomy and how do we actually name things or classify things. And we talked a little bit about Aristotle and how Aristotle broke every living thing down into two main groups, plants and animals. But we know now that things are more complicated than that, and there are a lot more different things. And from those characteristics, we were able to name those organisms using those 7 levels of taxa: kingdom, phylum, order, class, okay, all of those, all 7 of those. Remember that a way to remember that was King Philip came over for green soup, having to remember those 7 levels. But how did we figure out what belongs to those 7 levels? So we started with Aristotle, but then we moved into the modern classification system, which is based off of what Linnaeus developed. And what he did was he actually looked at things that were a little more specific than Aristotle, not just plants and animals. One of the first things that he looked at was the morphology of those living organisms. And that morphology means some shared physical characteristics. So the examples we have would be for an animal, what type of heart do you have? We have what's called a 4-chamber heart. Other animals have 2, other animals have 3. Okay? Another thing to look at is the type of blood they have. Okay, insects have something known as hemolymph. It's very different from our blood that's red, that has red blood cells in it. It's made of different things. We could also look at, do they give birth, live birth? Do they lay eggs? Do they have antennae? Do they have wings? So here in morphology, you're looking at physical characteristics. The same thing can actually be done for plants. So plants are also living things. They also have very specific scientific names. And so for plants, you could look at things like their male parts and the female parts. What are the reproductive organs? What type do they have? In addition to looking at morphology, that physical aspect, we can also look at the biochemical aspect, which is your DNA and your amino acid sequence. So biochemical analysis, this should be very similar to you because we've seen this before. The other thing that we can look at is embryology. Embryology is what is the common fetal development. So if you want to take a minute and kind of pause and look at this picture that we have here, here are 3 or 4 different organisms: a lizard, a tortoise, a pig, and a human. And here's what they look like at different stages in development. And notice at the very beginning there's lots of similarities. And these similarities start to get a little more and more different as the cells begin to differentiate more, as more mitosis occurs, and more cells are being accumulated. But those are 3 major things that we can use to classify organisms: morphology, the physical characteristics, biochemical analysis, which is DNA and amino acids, and embryology. How do they develop to become more complex? Another thing that we can look at is phylogeny, and this describes the evolutionary relationship between organisms. Notice that in our example, it says we'll look at homologous and analogous structures. Phylogeny means we're looking at what connects those organisms, and a lot of times it will be those physical bones and wings that we can look at. When we're looking into phylogeny, we'll look also at phylogenetic trees. Phylogenetic trees, this is an example right here, are used to show a visible representation of the evolutionary relationships. So in this picture, you can see at the very bottom, we have some sort of starting cell, the first eukaryotic cell possibly. Then from there, we have things that are like fungus and animals. Fungus are very closely related to animals, not that closely related to plants. So we have a connection there at this point. From there, the animals become more complex. You have things like insects that have an exoskeleton, hard outer shell. You have things like vertebrates, which have a backbone, like us, like cats, like dogs, like turtles. Okay? And from there, there's even more differences. So for a phylogenetic tree, each node, each connection point you see here is going to refer to a recent common ancestor, and then there was something that caused them to split and change. Something that we have that's similar to a phylogenetic tree is a cladogram. It's a specific type of cladogram that is actually going to emphasize the order in which the derived characteristics arise from a phylogenetic tree. What does that mean? The order is really giving us a time frame. So it gives a time frame. What they're saying is that one characteristic came before another. So looking down here, you have these different types of some aquatic organisms. And here they show you that, okay, 4 limbs evolved here. Well, this happened after we already developed our ray-finned fishes, like stingray, and before we had our after we had our sharks. So sharks came first, ray-finned fishes came first, then we developed 4 limbs. And from the 4 limbs, then we can have our amphibians, turtles, lizards, snakes. So all of these organisms that are in the shaded section have our 4 limbs. So again, a cladogram is a type of phylogenetic tree, but it's going to give us some time references. What happened first, what happened last. From there, tools used in classification, not only do we use those phylogenetic trees or cladograms, but we can also use what's called a dichotomous key. Okay, I'll say that again. A dichotomous key. A dichotomous key is almost like a little word search. You're actually going to use paired statements and visible characteristics to key out or to find, identify a specific organism. I'll show you what that means. Here in your notes you have a picture of a leaf, and this here represents your dichotomous key. So this is a dichotomous key. One thing with a dichotomous key is you always have to start at number 1. Even if I had 5, 6 pictures, on the 6th picture, I would still start at the top of this key. Looking at this leaf right here, what I would do is I would look between choice 1A and 1B. Is the leaf divided into leaflets, or is the leaf not divided into leaflets? Leaflets would mean that you have little small leaves like this. Okay. Does this have leaflets? No, it doesn't. So it is not divided into leaflets. So what I would do is go to step 2. Step 2, I would also look at A and P. So take a second to look at A and B. Which one matches with this example leaf? Well, A says veins branch from a single point on the leaf, or veins branch from a main vein in the middle of the leaf. The veins means this, those lines, are they all coming from the same spot? Okay? Well, they're not coming from a single point like this one, so they're actually going to come from a main vein in the middle. Look at those little veins branching out. So it says to go to step 4. Going to step 4. The leaf has a jagged edge, or the leaf has a smooth edge. Well, if you're looking at this, this is very jagged, right? It's not rounded like this, not smooth. So this is actually going to be a birch leaf. That's the dichotomous key. Hopefully it's pretty simple, right? We're just going through step by step. If I had another leaf like this one, I would also go back to number 1. Once I finish identifying this, I'd go on to the 3rd picture, but still start at number 1 each time. And you're going to have lots of practice with this later on. And that's going to be it for today. For your summary box, go ahead and take a minute, write 2 sentences summarizing up Unit 4.3, and that's all of 4.3. What are 2 major things that you know are really important? What 2 topics do you expect to definitely be on our test or quiz? So write 2 sentences in our summary box, and we'll check that when you come back.